In this work, we investigated the effects of reagent introduction order for K2YbF5 synthesis with the name of downsizing our crystal in order to use it effectively for biomedical application. For the first method, we add together potassium hydroxide, oleic acid, and ethanol, followed by lanthanide ions, and then finally potassium fluoride. Then we add in, for the second method, aka the separate solutions method, we mix the lanthanide ions, oleic acid, and ethanol. And then we follow it by potassium hydroxide and then finally potassium fluoride. Under the third method, which we call the together solutions method, it's similar to the second one except that we combine potassium hydroxide and potassium fluoride together first before we introduce it into the system. Data analysis of XRD peaks show that the unit cell parameters agree with reference data except for the crystals made under the together solutions method, and so cast doubt that it is in fact K2YbF5. Then we looked at the SEM images, and it seems that these SEM images agree with the XRD data. And based on the SEM images, we don't really see the expected downsizing of K2YbF5 crystals as we see on the separate solutions method crystals, the width increased, and for those made via together solutions method, we see that the morphology changes instead into micropolyhedrons. So the proposed explanation here is based on how much of the precursor ions and how much of the expected reagents get encapsulated into the micelles that form as the micro reaction chamber essentially, and based on that we derive our explanation on it. And according to our next steps data, we show that we do have erbium doped into our crystals, and both ytterbium and erbium have the required energy levels for us to generate upconversion. And according to our photoluminescence results, we see that we get green and red emissions for the crystals made under the together solutions method, whilst we only get red emissions for those made under method A, the original method, and the separate solutions method. And so based on this, we can say that the emission of those made via together solutions method is the best amongst those tested. However, given the sizes of the crystals that we have here, we can't really say whether these particles will be suitable for biomedical applications.